بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. Our lecture is about the structure and functions of RNA. There are many objectives of this lecture. First, define the structure of RNA. 2. Contrast RNA and DNA. 3. List the types of RNA and explain their functions. 4. Explain the process of transcription. Explain how the immature messenger RNA modified and spliced. What are the differences between DNA and RNA? RNA is a single-strand molecule closely similar to molecule of DNA, but differs from the DNA in that single-stranded rather than double-stranded. Contain ribose sugar instead of deoxyribose sugar. Contain uracil instead of thymine. RNA is single-strand polynucleotide molecule and have a lot of the RNAs types: messenger RNA, tRNA, and RNA. Functionally, DNA stores genetic information, whereas RNA controls how that information is used. There are three types of RNA: 5% messenger RNA, 15% of tRNA, and 80% of ribosomal RNA. At the first, messenger RNA is about 5% present in the cell. It is a linear sequence of the nucleotides formed in the nucleus. Carries the genetic information copied from the DNA in form of 3 bases, each of which specifies for a particular amino acid. It is passes from the nucleus to the cytoplasm, where it is found association with ribosomes to serve as a template for the ordering of the amino acids into the polypeptides. So the information flow from DNA to messenger RNA, then into the polypeptide or called protein. For example, a muscle cell has many messenger RNAs that specify the contractile proteins actin and myosin. And the other example, skin cell contains many messenger RNAs that specify the protein keratin. To use the information in the messenger RNA sequence, a cell requires two other major classes of RNA, that it is ribosomal RNA and tRNA. In tRNA or transfer RNA, also called adapter RNA, because it is a much smaller molecule of RNA. tRNA is made up of single strand of RNA consisting about 75 to 80 nucleotides, twisting and folding upon itself to form double strand regions. At the one end, a single strand loop at one end of the folded molecule contains a special triplet of the bases called anticodon. This anticodon complementary to the codon that's on the messenger RNA. At the other end, contain specific sites for the specifying of the amino acids. What are the functions of the tRNA? It is the key that reads the code on the messenger RNA by the anticodon region complementary to the codon on the DNA and picking up the appropriate amino acids which have been activated by special enzymes. This site occurs at the other ends of the tRNA, specialized for the picking of the amino acids. The third type, ribosomal RNA. The ribosome consists of two subunits, large and small subunit, each of which made up of protein and ribosomal RNA. The larger ribosomal subunit has three types of ribosomal RNA as well as protein, and the small subunit has one ribosomal RNA as well as protein. Each ribosome has binding site for the messenger RNA on small subunits and two binding sites for tRNA on the large subunit. The first site is B site. B site specifies for holds the tRNA carrying the growing polypeptide chain. Another site, A site, holds a tRNA carrying the next amino acids to be added to the chain. What are the functions of the ribosomal RNA? Ribosomal RNAs catalyze the formation of the peptide bonds between amino acids such as ribozyme. Also help to align the ribosome and messenger RNA. What's the meaning of central dogma? Central dogma, that means the transfer of the genetic information from DNA into the RNA by the transcription and from the RNA into the protein by the translation. The process of transcription occurs in three stages: initiation, elongation, and termination, as in replication process. Transcription begins at the first step, RNA polymerase and transcription factor bind to the specific nucleotides sequence in DNA. This is called promoter region. Promoter region, which marks the beginning of the gene. The adjacent double-strand DNA unwind and exposing the template strand. The nucleotides that will make up the messenger RNA molecules form H bond with complementary nucleotides. In elongation stage, RNA polymerase joins the RNA nucleotides into the polynucleotide chain. The rules of the base pairing in the transcription are the same as in the replication, with one exception. Adenine on the DNA template specifies for uracil in the RNA transcripts. That means there is no T or thymine in the RNA. In a human, there are 30 to 50 nucleotides per second are added to a messenger RNA molecule. At the last stage, when the RNA polymerase reaches to the end of the gene, that it is called terminator region. It detaches from the DNA template and the messenger RNA is released and the DNA strand reform a double helix and the transcription is terminated. In a human, most of the transcripts are about 5,000 nucleotides, maybe reach up to 200,000 nucleotides. How can differentiate between replication and transcription process? There are main differences between them. One of these, RNA polymerase enzyme instead of DNA polymerase enzyme. RNA polymerase attach to the region called promoter region in transcription, whereas the DNA polymerase attached to the origin of replication in the replication process. The base pairing are the same in both replication and transcription, but transcription differs from the replication by uracil presence instead of thymine. There is no thymine in replication. Messenger RNA is processed and spliced. The original transcript from the DNA is called pre-messenger RNA. Both genes and pre-messenger RNA contain nucleotide sequence that are transcript but not translated, that it is called non-coding sequence, called introns. This can vary from 0 to 75 nucleotides. The other types of the genes or sequence that it is called exon. It is called coding sequence. That means is transcripted and translated into the polypeptide chains. The combination between exons and introns form of the length of the gene. نلاحظ أن معظم الجينات بالإنسان بالإضافة إلى البري ماسنجر RNA النسخة المستنسخة من الجين تحتوينا على نوعين من النيوكليوتايدز. عندنا النوع الأول يسمى بالنون كودينج نيوكليوتايدز اللي هو الانترون، معناته هذا يستنسخ ولا يترجم إلى بروتين، بينما النوع الآخر هو الإكسون، هذا يسمى بالكودينج سيكونس، هذا يستنسخ ومن ثم يترجم إلى بروتين. بمجموع الانترونات والإكزونات الموجودة على الجين أو البري ماسنجر RNA تكون الطول الحقيقي للجين. At the first, the transcription produce large RNA that it is called pre-messenger RNA, contain exons and introns. This premature or pre-messenger RNA cannot move into the cytoplasm and associated with a ribosome for the translation process before become mature. That means the pre-messenger RNA should be processed and modified in the nucleus to produce mature RNA. The processing and modifying begins with capping. The first step, it is called capping. In capping, addition of the nucleotides, that means methyl group to the guanine, to 5 ends, that called cap, play important role in the aids in the binding of the messenger RNA to the ribosome during the translation. Another step, addition of polytail, a tail, at the 3 ends, adding about 30 to 100 adenine nucleotides. at the three ends, that it is necessary for protein synthesis to begin and for stabilizing messenger RNA. At the last step, that it is called splicing, all the introns are removed and the exons are spliced together by special enzymes called ligase enzyme to produce mature messenger RNA. After maturing of the messenger RNA, the messenger RNA moves to the nucleus, from the nucleus to the cytoplasm, readily associated with a ribosome for the translated into the protein. Several human genetic disorders are a result of abnormal premature RNA splicing. In beta thalassemia, one or more mutations at the border between introns and exons that lead lower the efficiency of the splicing and as a result cause deficiency in the amount of the gene product beta globulin synthesized and cause genetic disorder called beta thalassemia.